of people started. Let's get going. And
okay, sure.
I want to start by acknowledging that the RC Center of Excellence for the Mathematical Analysis of Cellular Systems sits on the land of the Wurundjeri, Bunurong, Turrbal, Yugara, Betgal, Gadigal, Ganabal, and Gambi people. And we want to acknowledge their traditional ownership and the custodians of the land on which we work and live. And I want to thank profoundly Markus Covert from Stanford to talk to us today. Markus wrote a paper in 2012 that I thought, and I know that you showed us something with Francis Crick. Francis Crick called his autobiography What Mad Pursuit. And I thought the paper that Markus published in 2012 was a mad pursuit. And in 2014, I had a Damascus moment running through Hyde Park when I thought this was just not insane, but insanely good, and we should all try to do this. So I've read the paper, reread it, and I've read and reread Marcus's Scientific American article where he goes through the trials and tribulations that gave rise to the Cell paper, which is just beautiful prose and a really nice tracing of this intellectual journey. So Markus has really been the sort of the person who fulfilled a dream that I think Francis Crick and Sidney Brenner first voiced in 1967. And I feel hugely sort of privileged that we are able to have him for the seminar, talk to us about his project on E. coli modeling. And yes, Markus, I think with this, without further ado, please entertain us.
Thank you. And such a generous introduction. It is true. I was thinking about Brenner in particular, much of the time when we were doing that 2012 work. And I will tell you, Michael, like one of the all-time highlights of my career, before we published that work, it was really important to me to talk to Sidney Brenner about it. And so he grilled me for hours in La Jolla about this, about that original 2012 paper. And then finally he thought that we could move forward with it. And that was something I'll never forget. So that's thanks for reminding me of that great moment. Okay. What you're looking at here, I don't know if any of you are fans of the modern artist Andy Goldsworthy, but he is a fantastic artist, and what he's famous for is kind of doing art in situ. So he'll go for a hike, for example. You should Google him. In fact, if I would forgive you for even Googling him during this talk. He'll do these incredible works of art in nature using only what he can find, and they're very temporary, of course. There's a beautiful documentary about it called Rivers and Tides, which I also highly recommend. It's really wonderful. Inspired by that, I actually asked a friend of mine who's an artist, that's Eric, who's in this picture. I asked him to go into my lab and, inspired by Andy Goldsworthy, to take what he could find and make us something that would be a way of thinking about whole cell modeling. So what you're looking at is E. coli, and but this is not photoshopped in any way. This is just a photo of filter tip pipettes and conical vials and cryo vials and cryo tubes, but at the same time of computer cables and computer components. And so what this is supposed to be about is bringing together experimental and computational components to build a model of E. coli. So that is what this is about. That's what the art is about. That's also what the science is about. That's what we're trying to do, bring together all of the data and all the things that are known and use computational techniques and metaphors to be able to understand how E. coli works at a system level. And as was noted, it was first articulated 50 years ago now by Francis Crick and an uncredited, just an acknowledged Sidney Brenner, calling for the complete solution of E. coli. And there were a number of really interesting recommendations in this paper. It's truly foundational. Almost none of them were followed, but it was visionary for its time. And I still think a lot of those things we would benefit by doing in E. coli. So inspired by this and many other things, we set out to do something like this. And what we're doing in particular is trying to take all of the knowledge that has been generated about E. coli over many decades. You're just seeing here, papers are rising over time. And we're trying to get all of those data now, hundreds of labs, thousands of papers, millions of data points. The data are heterogeneous, as you see here, just from the size of the data set in each paper. How do we bring that all together? And the answer is we can encapsulate it in mathematics, right? So we can take the parameters, write these equations, let's say 4,500 equations for every RNA in the cell. That incorporates a set of parameters. We can do the same thing for the 1,500 or so small molecules. That incorporates a whole nother set of parameters. We can do that again for the proteins in the cell. And now we've incorporated those parameters and can interact. Now, if we group them by their functional categories and kind of apply our modeling special sauce, we have a way of taking all of this knowledge as a whole and starting to look at what happens to overall behaviors in the cell. That's the key goal of this project, is to try to enable previously kind of impossible discoveries, because we're looking at the system in its entire context. And once we bring all of those things together, we can start to run simulations that are surprisingly lifelike. I've just been talking to my lab about this. Some of you will know the Turing test, where Alan Turing proposed that, a robot, basically, if a human can talk to a robot on the telephone and not recognize that they're talking to a robot, that starts to sound like it's passing certain tests of intelligence. And as I show you, as we stimulate these cells with different environments and their gene expression shifts and sensors are triggered and they double at different rates, we're starting to generate data, and you'll see this towards the end of the talk, that is really starting to approach a remarkable situation in which I believe if a person came to me now rethinking Turing's test, but in the context of a cell and molecular biologist, if you were to come to me and ask me to see my data about E. coli, I think it's pretty likely that I could come up with a data set that it would be extremely difficult for you to prove was built not by artificial intelligence in this case, but also not by experiment, by an artificial life, by artificial simulation, right? By a simulation that's simulating the life cycle of E. coli at scale, and with the full kind of a large amount of the actual biology and chemistry that underpins it. So we reported in Science just a couple of years, 2-3 years ago now, I guess 3 years ago, the production of what we call the half cell model of E. coli. It accounts for about half of the annotated genes and their functions. We are now busily working toward what we call the E. coli whole cell modeling project, which is we're hoping that in the next 5 years, we will have 90% of the annotated genes functionally represented in our model and be able to perform a wide variety of interesting simulations on a wide variety of environments, and be able to make realistic simulations that can give us insight into how E. coli actually behaves. And I'm just showing you a run through here. I could talk about any one of these stories. I'm going to talk about 2 of them today. The testing key predictions here at the top, and then the new functionality, the tRNA metabolism. I'll talk about those 2. If anyone wants to talk about anything else, obviously, more than happy to. So first, let's talk about complex phenotypes, and we refer to this a little bit in our science paper. So I'll use that as a springboard. And I want to, of course, thank all of the amazing people who have worked on this. This is I would say this project, more than anything I've ever been a part of, requires an incredible amount of teamwork and discipline to be able to work together as a team. And of course, that's not necessarily an academic strength, right? So we've really had to learn to be generous with each other and bring our strengths together and recognize each other's gifts, while also coming up with our individual contributions. It's been a real challenge, but I've loved it, and all of the people working on this have been phenomenal. Okay, so maybe just to get into this, I want to talk to you about a hierarchy of model predictions. And it really goes back to my own history doing modeling, starting as a graduate student. You know, when I started out in Bernard Paulson's lab, the greatest thing I could possibly imagine was to be able to make a prediction with my model that was able to kind of recapitulate or send back to me something that I had withheld from the construction of the model. So previously measured, you know, the data set existed. I just removed a little bit, and I was able to then use the model to kind of give me a very good guess of what I had taken out and what the values were. So I call that validation here, just for simplicity. We could talk about what these different things mean, et cetera. But for now, just when I talk about it, that's what I'm thinking about. When that first happened to me, I could not believe it. I was so excited. I thought it was the greatest thing ever. And then, you know, that kept happening, and I started getting excited about doing my own experiments, right? So I remember actually trying in San Diego to get somebody else to do an experiment for me, and they said, why don't you do it yourself? And I thought, yeah, why don't I do it myself? So I had to take, you know, an extra year just learning techniques, et cetera. But the thrill of making a bona fide prediction with the model that I knew had not been tested before, and then doing that experiment and finding something comparable and realistic was about as exciting as anything I could imagine at the time. Great way to end my thesis. And I'll call that discovery, because it's something now that we didn't know, right? And so in the case of whole cell modeling, you know, I would say the whole cell model has now been compared to about as much data in a of E. coli, right? Has been compared to about as much data as you could reasonably hope, right? We've compared it to all kinds of different data sets. We'll continue doing it, but it's remarkable to me how often the model gives us back a fairly good estimate of things that we're purposefully removing as benchmarks. Similarly, the model, in much more limited, but the model, whole cell modeling has been used to predict kinetic parameters correctly that have been tested later, predict protein degradation rates that have been tested later, and that's been very exciting as well. So now I'm hungry for the top of the pyramid, which is to say, I really want to be able to identify large-scale complex phenomena that no one has seen before, and using the model as a guide. And so I tell my lab, this is, of course, we don't set our sights on prizes. We set our sights on just being useful to the world as much as we can. But I tell my lab as a motivator, you know, there certainly are at least 10 more Nobel Prizes hiding in E. coli, right? So there are, CRISPR was in E. coli, right? Like there are incredible insights just waiting to be found if we have the ability to get after them. And so that's what I'm hoping, that we identify these like large scale, interesting new insights into how E. coli works. And here I have to excuse my dog, who I think just recognized my daughter got home from school. Okay, so thinking about these new paradigms, that's where I want to go right now. So even in that first science paper, there was a new paradigm that was identified, and it's kind of like all of them. You'll see as we keep going here, it's kind of complicated. So I want you to take a look at this and just settle into it a little bit. So if you're new to this kind of modeling, so on the left in red, we have the expression of a gene, okay? And it's 6 generations. So if you look at the protein count, the upper left, you're looking at the protein count over time in a single simulated cell over 6 generations. So each one of those red lines going up is a single generation. And what happens is pretty much what you would expect for this RNB gene. The protein is being produced, and then the cell divides. And so the protein count in the new daughter cell is half. Okay, and then the protein is made and expressed over time. The cell divides in half again, and it does this 6 times. If you look on so hopefully that one's somewhat intuitive, right? And it's going in roughly an exponential, growing in roughly exponentially. And so we call this actually exponential expression of the protein. If you look down at the mRNA, it's a little more surprising for people, although this is the kind of thing people have measured in individual cells. The mRNA is much more oscillatory, like fluctuating, let's say. It's a much lower count. And the reason for this is that the mRNA half-life is so short. It's just a few minutes, right? Whereas the typical protein half-life in E. coli is many hours. And so you have something that looks more stable at the top left and something that looks much more random and stochastic at the bottom left. None of this is maybe the key thing for you to know here, now that I've just explained what I see when I look at this, is that none of it's really surprising, right? This is actually, what you're seeing on the left is exactly what I expected to see for all the genes, and it is what I see for many of the genes. But I see for most of the genes, and this was probably the single biggest surprise for our first round of simulations. What I see for most of the genes looks more like what you see on the right. And let's look at the right bottom for a second. So here, over 6 generations pass, and only one mRNA is produced from this gene. Right? It's produced only once in 6 generations. It's a very low expression, but when this happens in a single cell, that still means that a discrete event has happened. It's not that every cell is producing 0.0007 of an mRNA, right? Like this cell is producing one of these mRNAs at this time in this moment. And as a result, when it is expressed, now go up to the upper right, when it is expressed, there's this burst. Not a burst. Sometimes people in physics talk about a burst as being something that involves regulation. So don't think about it that way. But there's just a sudden increase in expression, of course, because you've gone from 0 to 1 mRNA. So now translation can occur, and about 150 proteins are made from this single transcript before it decays. And then what happens after that is every time now that the cell divides, each daughter gets half. So you're looking at these little decreasing height stairs going down, right? Because the cell divides and there's half of the protein, and then it divides again because nothing new is being produced, and it divides again. And so it goes down in stepwise in this really interesting fashion. So when I saw that for just one gene, I was scratching my head over it. We call it now subgenerational for exactly the reasons I just explained. One transcript, many generations. But it turns out, here what I'm showing you is a plot of all the genes, okay? And they're ordered by how often they're synthesized. And the red ones, it's kind of the same as here. The red ones are exponential, and you can see that they're about 1/3 of the genes are expressed exponentially. So it's not that they're uncommon. But look at this, 60% of the genes are being expressed less than once per generation at the mRNA level. Now, it's even true that there's a significant fraction for which there's no protein sometimes. And it's even the case, if you want to get even crazier and more mysterious, it's even the case that some of those proteins are essential, and yet they are not always present in the cell. And the cell can survive that. I could talk about this all day. Unfortunately, there's no time. It's really fascinating to think what the functions are of these subgenerational genes. Maybe I'll take a second on that. Because when you look at what these things are doing, like a very large fraction of, for example, the antibiotic resistance genes are subgenerationally expressed. And for me, this was the biggest clue of what was going on. Which we kind of use a bet hedging metaphor. Or sometimes I just think about it as E. coli's suitcase. You know, you'll see other people talk about E. coli as this incredible computer or this incredible metabolic engineer. I think of it for these purposes as a suitcase. It can only hold a certain amount, right? So it can't hold all of the expression of all the genes it would need to be ready for everything. And so I think the suitcase analogy is great, because if you're going to, let's say that you're going on a vacation, but you don't know where, right? How do you do that? How do you respond to that? Because you can't fit your skis and your ski boots in the same suitcase as your surfboard. And similarly, what happens is E. coli can't fit resistance to every single antibiotic, plus the ability to respond to carbon sources that it doesn't see that often, plus these other abilities that it needs, it can't fit them all in that one little bacterial-sized suitcase. And so as a result, what happens is, instead of everyone in the colony being ready for anything and everything, everybody in the colony, let's say, is ready for one thing or some small number of things. And that way, let's say that there's a whole colony and one of them just had that expression event. They are now resistant to some antibiotic. Okay. So the antibiotic comes through, everybody dies, right? Then boom, divides. The concentration in each starts to reduce, right? Divides. Now, none of these cells have an appreciable amount of the protein left, but there's been another expression event, and now this one has it. Do you see? So the colony retains the ability to respond to everything, while each individual has a much more reduced ability to respond to changes in the environment. That's how we interpret these data, the subgenerational data. It's a way of having bet hedging and enabling cells at the colony level to be ready for anything. So one of the fun things about what we do these days is we have this partnership with EcoPsych. And so if you ever want to look at kind of exponential or subgenerational genes, every gene in E. coli now, there's a modeling tab. If you look just right in the middle of this slide, there's a modeling tab that you can click and it will tell you the count or concentration per cell. So you can actually start to get an idea of, and you can compare that to experimental data. We're starting to make this automatic pipeline where EcoPsych changes are immediately read by the model and incorporated, and model predictions are automatically returned back to EcoPsych itself, so that people can benefit from the simulations and not just have to know the code and all that stuff. Subgenerational sounds like someone else from the family is coming home. In fact, I think I'm gonna take 1 second to close these doors. I'll be right back. So as I was saying, it's a fun prediction, and like I said, this is one of these cases where it's kind of a paradigm shift, but is it actually correct, right? Does it mean anything? And the answer is, we have to find out by testing. So if you look at this slide, what you're seeing is 3 genes, these 3 columns, the 3 leftmost columns, LAC-Z, GCFE, and BAES. Those are all genes that the model predicts should be subgenerationally expressed. And then on the right there is HUP-A, which is a gene that we would predict to be exponentially expressed. And so you can see these first 3 rows, those are simulations showing, look at the second and third one, right? They're showing the expression of an mRNA. And then they're showing the protein that results in that characteristic like downward staircase that happens after as the protein's diluted out. Now we actually made those constructs in chromosome fusions to M. venus. And so you can look here at the colony level, which is kind of fun. So you see these little islands of expression, which is what you would expect to see in the case of a subgenerational gene expression. And HUP-A, of course, everything has the same intensity, the same expression. But it gets more exciting here when you can look in the microfluidic domain. So here we've got cells at the bottom. You can see all of the cells. They're expressing a constitutive plasmid here. At the top is LAC-Z M. venus. And so this is a mother machine, which is to say they grow in these troughs. So you can see one cell for a long period of time. And if you're looking, you should be able to see these little flashes like that of subgenerational expression. So you can actually watch these things, because unlike a colony where things get crowded and it's hard to track things, here in the mother machine, you can watch, you can quantify, you can segment the image pretty easily. And so that's really exciting. I'm just seeing a Q&A. Do real observations update the model parameters? Yes. So I would say real observations are used in most cases, but you'll see a couple of cases where we have to go forward, right? So I'll talk to you about that in a minute. So the answer is yes and no to that. Okay, so back to the mother machine. Hopefully you saw some of those flickers. We can take them. Here is in the middle. So on the left is our prediction, which I already showed you. In the middle, you can see that mother machine, and on the for each time point, you have the constitutive reporter at the left and M Venus at the right, and you should see that little flicker in the M Venus channel, which we can quantify over time. And to me, this is why I do science, right? So here's the prediction on the left. There you see the characteristic downward staircase experimentally in an individual cell at the right. And to me, that's the stuff dreams are made of. And I'll just say, maybe to go back to that for a minute, we're now starting to look at this in operons. We have a number of things we're thinking about. We've made several constructs. I love to work with my own hands in the lab during the summer. So I've actually made one of these constructs, which was exciting. So yeah, that's a story that's on its way and in progress. So that's a paradigm shift where we have some data. I wanted to show you that as just something that came out of something you might have seen if you saw this science paper. Now I'm going to show you something that was just published earlier this year. It's one of the most exciting things I think that's come out of the lab, but it's all computational. In fact, I'm desperate. We just hired 2 new experimentalists, and one of our goals is to try to figure out better ways to try to test what's coming out kind of this really interesting work. I have to thank Hee-Jo Choi here. She did almost everything herself, and it has truly been amazing to watch. She is incredible at kind of detangling very complex mechanisms. So I talked to you about Francis Crick. Now I'm going to talk to you about another 40-year-old paper. In this case, it's a 40-year-old controversy, right? So this paper was published, and there are surprisingly few follow-ups, really almost no follow-ups to this paper, even though it makes kind of an astounding assertion. So they actually went in and measured individual amino acid tRNAs and how fast they turned over. And the thing that was really important was they took kind of all the rate, they gathered all the rates that people had measured, and then they compared it to what they thought E. coli would need to grow and divide. And they made this astounding assertion in their paper at the very end, which I've highlighted here. Basically, they say that their estimates of what would be required in vivo is much, much higher than anything that had been measured in vitro. Okay, so they basically are asserting here that the in vivo measurements, that the in vitro measurements are not anywhere close to the in vivo measurements for whatever reason. 40 years ago. And nobody really was able to challenge that because it's just very difficult to measure these activities at all, and not even to mention in vivo. But what's kind of incredible about a model like the whole cell model, often we just talk about predictions, but what's really cool here is that we can actually start to think about simulating the in vivo context, right? We have a model of the in vivo context, so why not use it? Why not try to figure out what those in vivo rates should be, and then go back and compare them to these in vitro measurements? So that's what we did. Hee-Jo went in and updated our model in a number of ways. The most important way here that I want you to see is that it's now codon specific. That turns out to be important later. This is kind of how we do all of our work. We find places in the model that previously were low detail, and we make them high detail. And we go look at all the data that's needed. So the new model is much better in a variety of ways. It gives you this idea of amino-isolated versus unamino-isolated tRNAs. It can tell you the difference of codon usage. It has a number of really interesting advantages over the original model. And so when we went to try the Goldman-Jakubowski assertion, right, all we did was we took our original model that didn't include all of the turnover rates related to tRNA synthetase, and then we added all those rates, and we wanted to see if there was any difference in the simulated doubling time, and you can see there's a huge difference. So on the left in black, you can see the actual experimental measurement, 44 minutes. Then you can see what the previous model was doing, kind of around the high 40s. And then you can see what happens when we add the in vitro measurements. It quickly goes, more than threefold higher at the mean. So from my perspective, this is theory-based evidence that the data, or that their assertion, is correct. And we can show, furthermore, that this is related to the tRNAs specifically and to those K-cats, because we can look at the terms that guide the amino isolation rate. And if you just take this is the full equation. If you take those three terms and you just assume that they're at their highest, right? Basically, the rate of amino isolation is going to be primarily set either by the enzyme concentration or the K-cat. And so this is then where we can start looking at other data. If you look at the proteome, which was not used to parameterize the model, if you look at the proteome, I've highlighted all the tRNA synthetases in purple. You can see that the measured and the simulated amount of the tRNA synthetases is remarkably, it's even more consistent than most of the proteome. So that would suggest that we're getting the enzyme concentrations right, by and large. And more importantly, even this now is the orange is the distribution that has been obtained from single cell imaging experiments. And so you can even get a distribution of what's going on with these synthetases. And even that is similar to what's being simulated. So this is very, very close. And so we are assuming that those enzyme concentrations are correct, which means that it's the K-cats that are the problem. And I'll just show you here, this is more on the validation and benchmarking side, but you can kind of see that we then, okay, well, actually, let me take a step back. So based on this information and this information, we decided to go and determine what we thought the in vivo K-cats should be. So this goes back to the question in the Q&A. We decided to figure out what those should be. And in doing that, it sounds like my dog is desperate to get back in. So 1 second. Okay, Bodhi. Once we made those predictions, we could take the resulting model, once we had predicted these new K-cat values, and see how well it was able to hit our typical validation benchmarks. And so in this case, it's the doubling time. Our current model actually is a little bit better than the previous model at hitting the doubling time benchmark. Although there's a significant amount of randomness, I wouldn't put a lot of weight on that. Proteome looks about the same as the old model. The fluxome data that we compared to, the agreement is about the same as the old model. tRNA abundances we couldn't compare to before. Now we can. We used those in figuring out the model, so that's not too surprising. But everything looks good. It looks like it's consistent with a lot of our benchmarks. So that was a basis on which we can start to run some more interesting simulations. You can actually look at the tRNA amino isolation in much more detail. There are several data sets, and none of them are very well consistent. If you look here, those are all the different colors, the reds and yellows and greens and blues. The model distributions are all in purple, and you can see we probably agree more with the entirety of the data, which again was not used as a to build the model in any way. We're probably more consistent with all of these data than any data set is with any of the others. So kind of a remarkable little figure here, but not really what the story's about. It's just to tell you we have some confidence that this model is going to give us interesting information about to get into Goldman and Jakubowski's work a little bit. And so now what we've done here, and this is the springboard, the real springboard for what we're about to do, we could go in, and what you're looking at in gray is the range of all the in vitro measurements, okay? And you can see specifics about the triangle is exactly where Jakubowski and Goldman thought that the lower limit had to occur. The blue is our estimates, our in vivo estimates, and the values we used in building the model. So you can kind of look at all of these interesting things, and I think it's pretty fascinating to go into actually why these different things are, why there's a difference. What's the mechanism of the difference? What is it that causes an in vitro? If there was, we can kind of look at this counterfactual. What if the value was the KCAT that had been measured in vitro? What does that actually do to the cell? I've highlighted two stories here. I'll refer you to the paper, which is in Nucleic Acids Research earlier this year for the first study, the histidine study. I'm just going to focus on arginine because that's my favorite one, and I think about it all the time. So if you look over here, first of all, just take a look at that green box around ARG-RS, the synthetase related to arginine aminoacylation. And you can see that there's somewhat over an order of magnitude between what Jakubowski and Goldman thought was the lower limit, and there's even more if you look at the in vitro measurements. So there's a pretty significant difference between what the model says and what had been measured previously. And so we can actually run two sets of simulations, one with our estimate and one with the in vitro measurement. And that is, in fact, what we're going to do. So the gray box, the gray and the leftmost is always going to be what we're calling like the wild type simulations, which means with the optimized in vivo hypothesized parameter for the KCAT. The purple is all going to be with the in vitro measurement. Okay, and so what you're looking at here is that with the model with the optimized parameters, the gray one, it's just running at that doubling time and it's pretty much staying there. Whereas if you change it to the in vitro, in this case, immediately the doubling time starts to go up and it continues going up. So it's not really stable. And you can see that at some point it hits 3 hours, and that's when we automatically halt our simulations. So these guys are failing. And they're failing after about 9 or 10 generations, but you can see the impact right away. And the beauty of a whole cell model like this, and something, by the way, that you would never see if you just did this all with AI, is that you can go deeply into the mechanism. And we call that deep curation, just kind of by way of connection to deep learning, right? So you're learning through different layers, you're peeling back layers, right? You're looking at interactions between interactions. And so here we're looking at the DNA, the major cell components, the DNA, the RNA, and the protein. When do those things gas out? And again, look first of all at the left. Normally it should just grow exponentially and then double, divide in half and grow again and divide in half. So what you see is that on the bottom, the total dry weight is what we knew before. You're totally failing at about generation 12. That's that green line. And if you look at the top, or DNA and RNA, they're also, RNA is failing at 12, DNA is failing at 11, protein is failing at 9. So that's the earliest thing to fail. It shouldn't be surprising because we know that these tRNAs are involved in translation. So of course, that's where we would expect to see something happening, right, in translation. So now we're going to dig back a little further. Okay, well, since it's translation, why don't we just go straight for the gusto and look at amino acylation? And sure enough, you see that whereas the amino acylation of all the other tRNAs is close to 100%, that arginine is down close to 0%. And that was fascinating to us because with the adjustment we were making, we were expecting something at about 80%. So this was our first clue. Like we knew we had knocked things down, but we definitely had not knocked them down that much. So what was going on with arginine? And so now we peel back another layer of the onion. We can go and say, okay, well, if the amino acylation isn't working, why don't we look at the amino acylation reaction and see what's going on with the substrates and products? And so what you see, if we look at remember, again, the gray is the optimized parameters, the purple or the bluish purplish is the model, the in vitro ones. And so you can see ARG-RS, that has not changed much. Arginine tRNAs is about at the same amount. ATP is about at the same amount. But arginine itself is down a lot, like a whole lot. That was also not expected. And I have these big arrows because we're on Zoom. I just want you to be able to see what I'm talking about. So then we can actually say, okay, well, why wasn't arginine there? We can look at this metabolic pathway. We can look at the fluxes through all the steps in the pathway. And so the first step starts with glutamate, and you start, and so you've got a concentration of glutamate. Doesn't look like anything's wrong there because nothing happens really to tank it out until generation 19. You can look all the way down at arginine, and sure enough, that has a major perturbation at 9. Ignore everything after 14. Those are just symptoms of a dying cell. But arginine is out at 9. You go upstream in the pathway upstream. Citrulline is still down at 9. And then you get to the N-acetyl transfer reaction catalyzed by ARG-A. That flux, if you look kind of second from the left, that flux tanks out at the 9th generation. And if you go to look at the expression of the synthase, ARG-A itself, pretty much the first step in the pathway, it is also reduced to essentially 0 by generation 9. Unexpected, you can see the gray there. None of that happens in what we're calling the wild type version of the model. So this was kind of a big new piece of the puzzle. If we then go to look at ARG-A expression, now peeling another layer down, you can see that the hexamer is gone at 9, sure enough. No complexation happens, no monomer is being made, no monomer is degraded. Because there's no monomer there, synthesis is not happening. So clearly ARG-A expression has been abrogated in a major way. Now we can do something, and this is where I'm starting to talk to you about these Turing tests, right? Everything I'm telling you is just simulations here, right? These are just simulations, but they're starting to really, I hope you see, take on a life of their own. And so we decided to do another experiment that that's quite complicated to do experimentally, and it's even not that easy in a simulation, but you can do it, and the data looks remarkably similar to what you would see in terms of just the look and feel. So now we're doing a ribosome profiling experiment. We want to see where pausing in translation occurs on the ARG-A transcript. Okay, and so if we look on the left, that's with the optimized KCAT. The in vivo KCAT, you can see there's essentially, especially for those arginine codons that I'm showing you, there's essentially no pausing. And if you look in the middle there, there's a uniform distribution that's saying essentially the pausing is happening equally everywhere. Whereas if you use that in vitro measurement, boom, look at CGG. All of a sudden, you have 90.4% of the time you have stalling at a CGG codon, which is a rare arginine codon. And that turned out to be incredibly interesting because if you now look at the ARG-A codon sequence, what you can see is that, in fact, there are premature terminations at the first CGG that occurs, 13.5% of the time, there's a premature termination at CGG. And then there's something even more exciting. There is a tandem codon repeat of this rare codon, CGG, CGG. 77% of the time, it stalls there. And there are CGGs after. Some people have asked me, but at that point, almost everything has already fallen off, right? So the presence of this dual rare codon was so interesting to me because here at Stanford, one of my idols is Charlie Yanofsky, who discovered attenuation in E. coli many decades ago. And attenuation is a fancy way of regulating amino acid biosynthesis at the translation level. And I don't have time to tell you exactly all the mechanism, fascinating as it is. It involves these kind of alternative hairpins. But the big thing I want you to see, if you look down kind of at the lower right in blue, you'll see UGG, UGG. You'll see that two, again, a pair of, hang on one second. It's asking for, hopefully that, can everybody see this? Hopefully you can see it. Stop me if you can't see it. So again, a tandem codon is regulating whether or not tryptophan gets expressed, which is fascinating. So immediately I started thinking about new modes of regulation via tandem codon repeats. So we looked at the whole, and now I see I've got to hurry, but I'm getting close to the end here. So we looked at the whole set of codons. We looked at all the proteins that in particular had tandem rare arginine codons, and there are several, but not that many, but there are several. And then we could actually look at all these different groups and we could say, okay, how is the expression changed for different proteins depending on what codons are in their sequence? And what we found was, now look at this blue arrow again, the proteins with CGG, CGG are significantly and about 20-fold reduced in expression from the wild type or from proteins that don't have any arginine codons. And none of the other groups are significantly different, even though they might look a little bit different in the fold change. They're not calculated as having significance. So this means clearly that the expression of those proteins, and I should just say, none of this was explicitly coded in the model. This is all emergent. Everything you're seeing here is emergent. So then we can do this other experiment suggested by our reviewer, who I would love to thank for that if they're out there. What if we replace CGG with a more common codon? We can do that fairly easily in the model. So that's a difficult experiment to do, but we can do it. And what we find is that sure enough, when you replace CGG, you rescue the phenotype. Okay, so you rescue the phenotype, and now you are able to produce arginine at normal levels or closer to normal levels. So what I've shown you here, hopefully, is that you can see that I'm kind of trying to trace this incredibly complicated system where you're going from an amino acylation rate and the fraction of amino acylated tRNAs. I'm showing you, if you look at that third row down, like specific places where pausing is occurring on a transcript, which is then translating to how much is produced from that transcript, the number of hexamers, the number of the flux through a reaction, the resulting concentration of arginine, and what that all means in terms of like ribosome termination, et cetera, and all the way down then to protein mass and eventually cell growth. What's really cool about this is that it establishes, I should say, an untested, unverified positive feedback loop in which ribosomal pausing on tandem CGG codons appears to have a regulatory impact on the concentration of arginine. And this is very fun in its own right. And I would love to hear anybody's thoughts on this because it seems to introduce a positive feedback loop. I'm just going to get the light here real quick. It seems to introduce a positive feedback loop that works together with the multiple negative feedback regulation that is already known in arginine. And for some of you out there, you might know that this presence of like what's called paradoxical feedback can have some very interesting properties as far as what that means for control of the concentration of this critically important amino acid. So with that, I'll just end here. The E. coli whole cell modeling project. Again, we're trying to get 90% of the annotated genes in the next 5 years. We were just funded by the Sloan Foundation to do this, and I'm absolutely thrilled about it. We have several other studies that have come out, and one on growth rate control that came out last year. This antibiotics one is now published in PLOS Computational Biology. The one on the left with operon structure is now in the second phase of review, and I expect that it'll be accepted in the next month at the latest. So we're starting to really put out interesting stories, and I'll just refer you only to the bottom right here, where we're simulating whole colonies and their responses to antibiotics. Like, again, thinking about Turing's test, I've worked with live cell imaging for a long time. And when I saw this colony, I thought, wow, this looks real, right? So I'm starting to feel like we're getting to a point where this system feels like a real system. And I've been thinking a lot about what artificial intelligence means and what artificial life means, what the benefits and advantages of each are. But I think artificial life had a really interesting history many decades ago. I'm kind of wondering if it's time to reboot and reinvigorate that field as we're starting to get to simulations that really do start feeling like artificial life. And what can they tell us in comparison with, but also in contrast with what artificial intelligence-based methods, which are also incredibly powerful and exciting, can do. So I'm very excited about that. We have a whole plan with all these things we want to do. I would love it. If anyone wants to join me, I would love it. We're hoping that people will come take some of these rows on. I'd be happy to share our plans with anybody, not just our code, which is all available, all our simulations, but also literally our plans, because if anybody wants to do something, we've hosted people and stuff like that. We'd love to help. With that, I want to thank the lab. They're amazing. Iran, up there, second from the right, is now just started as a new professor at the University of Connecticut. He's doing great. Couldn't do it without him. And so many thanks to them, and also to our funders who really have been amazing. It's one thing to have a mad pursuit, and it's another thing to have somebody who's willing to pour some money into that craziness. So I'm very thankful to them for taking a chance on a long shot. And with that, I'm happy to take any questions.
Thank you so much, Markus. I can't start my video because I think you're the host now.
Oh, here.
So if people please put any questions in the Q&A. Thank you so much. It was just a talk where I oscillated between smiling from ear to ear and just being open-mouthed at the results you can achieve. So let me ask, the first question is, starting with your last part of the talk, Jason Chin has done this amazing and astonishing work on reprogramming genetic codes. In principle, your model should now be able to adapt his esoteric genetic codes and predict life. Is that right? Or
that is absolutely fascinating. I think he had floated it to us one time, and we are thinking a lot about it. So everything so far with the E. coli whole cell modeling project has essentially been, we think about it as basic science, right? So we're just trying to figure out how the cell works. But I have a couple students now who really want to think about engineering and design. And so they are starting to add the software infrastructure for us to be able to make larger systemic kind of engineered changes. And so, yes, I would love to be able to do and show something like that. Yeah, I love that. That's a great comment. And thanks for the kind words.
Oh, no, it's easy to praise that talk. Any other questions? Oh, Ben, so let's have a look. Ben Hess, do you want to read them out? You can see them yourself, I think.
Sure. So Ben Hess is asking, given subcellular expression, how do you choose a starting state for a whole cell simulation? Is there something to be learned by starting the simulation from different states? Great question. So just like you'd be familiar with in other simulations, we run it out for several divisions. So we have a set of we have this whole initialization routine that brings in all of the experimental data and kind of recalculates everything. That said, it is interesting to start the simulation from different states, and more interesting from my point of view, we just recently added this feature where now kind of mid-simulation you can change. So for those of you who are interested in like causality theory or whatever, we're able to now do something with the model that you would never be able to do experimentally, but that's so fascinating, which is to actually run a counterfactual where you have a simulation that you run for 8 hours or whatever, but at 4 hours you put it in a different trajectory and you can see exactly what happens with that same cell, the same seeds, same number of every internal molecule. All the changes will happen from that instant. And so, yes, there's definitely something to be learned from doing that. I am fascinated with causality theory right now. I know nothing about it. I feel like I would love to figure out ways. Maybe just to add this, I told my lab this lately. I said, we're at an embarrassing time right now because even though Hijo did all of that stuff and it was amazing, maybe the dirty secret of that work is that it took her a long time. So we've gotten to a point where it's just as difficult to figure out the story on our simulations as it might be to do it experimentally. That shouldn't be the case. And actually, this is somewhere where I think Michael potentially has a lot to add, or anybody who does kind of data analysis, parameter estimation, these kinds of analysis type stuff, which is not our primary expertise. Because what we need now desperately is a way to analyze our simulations much more rapidly so that we see that huge pathway that took us months to figure out, so that we see it much faster. That's the limiting step. And it's still the limiting step for a lot of experiments. So long answer, Ben, but I love the question. Okay, so Malcolm, have you used your models to understand how energy draining futile cycles function in cells? The answer to that is interesting, and here I go straight to Sydney Brenner. So when I showed Sydney Brenner that we had a model of mycoplasma in 2012, he immediately started thinking of ways to confound me by just asking me questions about the simulations based on things he knew about E. coli that weren't widely known. And the very first thing he asked me was, how do you account for all the energy usage in the cell? And I said, well, it turns out that we can account for production, but like very little of the utilization. And he, this was, I think, the first time he took me seriously, because he said, wow, that's what I agree with. I think most of it is, wasting, like through futile cycles or other energy wasting. So it was the first time I started thinking about how hot, when we're growing cells in a batch culture, we're typically increasing the heat to raise the temperature of the culture. But I think in industrial applications, they're actually also cooling down because they are generating heat in mass. Anyway, I don't know the answers to all of those things, but it is fascinating. So I thought I'd just pass that down. Then let's see. Mark Wilkins, with your recent models, are you starting to consider interactions between cells? Yes. So what you saw on the last slide was the first ever whole colony models where you have a colony of 1,000 cells. They are all, in that case, interacting through the small molecules that they produce. But we haven't done much with intercell signaling. We haven't done much with, they push each other, but they don't really signal to each other by pushing or by interacting. So that's a big thing I want to do. It's kind of in those remaining, 30-35% of the genes left that we have to include. And I'd love one day, I don't know if you're familiar with Gurul Sewell's work, where he shows that bacteria can even propagate action potentials in a biofilm. That just blows my mind. So I would love to be able to simulate action potentials in a biofilm. but that's probably some years in the future. Okay, Carlos Lopez from Altos Labs. Oh, interesting. So here we are within just a few mile radius. Regarding the RNA observations from your models, many cells simply do not have mRNA for a given gene. Does that mean we have to revisit how we are collecting single-cell RNA-seq data? Usually we just throw away the 0 reads. Yes, absolutely we do. In bacteria, it's complicated anyway, right? Because you have all the... typically in a single cell, this is in a different side of my lab, but we were the first to come up with a technique in mammalian cells where in the same cell you could look at signaling molecule dynamics over time and also get the RNA-seq that goes with it. But even in a mammalian cell, you're getting 10 to 15% of the real RNA because of the dropout. If you were to count all of the mRNAs of every gene in a cell in a bacterial cell at a given time, that would probably be somewhere around 3,000 to 4,000 total. And so, right, let's say you're getting 10% of that or 15% of that. That's just, it's not even 500 copies. It's going to give you a very skewed idea of what's going on, not to mention the fact that every individual cell is going to look different. So that's fascinating. I'll tell you one other thing. I have a whole story about this, but in our Operon paper that should be coming out in just a few months in Cell Systems, one thing we discovered was through means which I won't tell you, what we discovered that the RNA-seq analysis tools also have a major limitation. And the limitation is that they tend to throw out all genes less than 100 base pairs long. And so the model, once the operons were included, the model was a better predictor of especially short genes expression than the data with the current analysis tools. So anyway, it just goes to show how when you include everything, like we throw away a lot, and some of it is absolutely wrong to throw away. And definitely the 0 reads is going to be wrong, especially in the single cell. And then the other thing that's going to be wrong is just short genes, which we actually went back to the raw data sets to show that these genes were being expressed at the levels that were roughly equal to what the model was predicting based on operon expression, but not equal to what the analysis was giving you, which was 0.
The subgenerational expression is a real eye-opener for me. I had not seen that. So in the boring question is, does that affect how you initialize the cell states? Do you really matter? Does that matter? And the more interesting question is how in your bed hedging approach, how much variability would cells have, and would there always be one cell in a population that would be able to persist or survive?
Yeah. It depends on the size of the colony, and it also depends on the gene. So every gene has a different probability of being expressed in the model. In our model, it's very stochastic. So we don't determine, even though I showed you ODEs, not everything is an ODE. So in our case, we decided that expression was best represented in a probabilistic or stochastic way. And so every gene has a probability of being expressed. And that's what leads to its expression across, let's say, a colony. Now, that said, there's a lot of regulation, some of which we know but haven't included, and other, of course, which we don't know, but at least we know that we don't know. We have an idea that there's stuff that people haven't discovered yet. So my assumption is that once a cell colony is kind of perturbed into a new environment, what happens is that there's feedback and regulation that then makes more cells able to thrive in that environment. So there's going to be a little bit of feedback that's also not included. But I think overall, what you see is just that these probabilities are giving you kind of this stochastic expression, which can kind of capture it. Maybe one other thing I'll tell you that's pretty interesting is this explains, we had somebody visiting who came and gave a talk, and and they weren't familiar with the simulation work, but they did a lot of work with antibiotics. This was Min-Soo Kim at Emory University. And what he had shown in his work that's fascinating is exactly what this model would predict, but what can be difficult to interpret if you don't have it, which is that he grew a lot of cells, he grew a lot of colonies in different wells of a plate, and then he would treat them with antibiotics, and maybe one well of the plate would still grow, right? And it's because they had expressed that subgenerational gene. But he was assuming that maybe that cell had some resistance. So then he would grow out the cell and plate it again in 96 wells. And then he would do the same. And of course, exactly what subgenerational would predict. Again, all the wells died except for one, but a different one, of course. But they all came from those same cells that previously had survived. So it's so yeah, I think there's a lot now behind this idea that bed hedging is a real thing, and that but most people, I would say, don't know the mechanism.
So one last question is to wrap up, and is also to give us ammunition when we talk to funders and colleagues. Is now the state where for E. coli in silico biology is more closely aligned to in vivo biology than in vitro biology is?
Right. This is an interesting question. I would say, maybe I'll say a couple things about this because I think it's really great. I tell when I give talks, especially when I'm talking to experimentalists, what I often say is, building a model is like playing two truths and a lie, that old game from being a teenager, right? Two truths and a lie, meaning that a lot of what goes in there is correct, but some of it is not correct, and you don't know a priori what's correct. You're making assumptions, so you know that those aren't completely correct. And I think experimentalists intuitively understand that a model has some truths and some lie based in it, right? But what's harder for them to wrap their head around, but I think which is underscored by this tRNA study, is that gathering data, especially in vitro data, is also playing two truths and a lie. It's still, you're still relying on unreliable narrators. And so I tell my students in class, my favorite riddle is, I don't know if you've seen this riddle with the two parrots, right? And each parrot is in front of a door, and behind one door is treasure beyond your wildest dreams. And behind the other door is certain horrible death. And one of the parrots always tells the truth, and the other parrot always lies. Okay. And somehow you have to figure out from those parrots, you're allowed to ask one question. You have to figure out which door you should go through. Now, what I love about this problem is that there is an answer, right? And the answer depends on the interaction between the parrots. Like one always tells the truth and the other always lies. So you can ask either one of the parrots, hey, parrot, like which door would the other parrot tell me to go through? And then you take the opposite door. So what I tell my students is like, what happens if we do 3 parrots or 10 parrots or N parrots, where M of them are telling the truth and the rest are not? Like, how can what's amazing to me about the 2 parrots is that even with unreliable narrator, you can get a completely 100% reliable output. And is it possible through modeling that brings together all these data, is it possible to take all of these unreliable narrators and somehow find the truth? And I think, this is the kind of thing that the philosopher should be thinking about, not me. But it is fascinating to contemplate. And so thank you for the question.
Thank you for the answer. It was just perfect. There was a frivolous demand in the room to see your dog, but I think we went over that. But it
is very cute. Hang on one second. I'll be right back. I'll satisfy that. Bodhi. Bodhi. Oh, hey, come here. Okay, there he is.
That's very well worth waiting for. Thank you so much, Markus. Thank you for this fantastic talk. It was just eye-opening, inspiring, and I just really enjoyed it. For those in the audience, John Glass from Crick Institute will speak next week.
Mm,
We're also looking forward to that. But Markus, thank you so much for your time, for introducing
of
us
course.
to your E. coli model and to your dog.
Thank you. It was great to be able to speak to you all. And thanks for those great questions. Let's connect soon, Michael.
Will do. Yes, I'll send an email. Take care.
Perfect. Bye.